Thank you very much, Ken. It's really a great pleasure to be here with you today to discuss vaccines and the state of the science. So to put in perspective where the state of the science is today, let us first consider where vaccine science stood only 10 years ago. So in 2001, genome sequencing was time-consuming, labor-intensive, and hugely expensive. With great effort, we had obtained the complete genome sequences for some simple organisms, including H. flu and a laboratory strain of MTB. That same year, the first rough drafts of the human genome sequence were published, and we still did not have genomic sequences for complex organisms like malaria parasites and the mosquito vector until the following year. Proteomics and other omics and systems biology were still in their infancy. At that time, we were just beginning to appreciate that pattern recognition receptors involved in innate immune responses might be key to understanding how adjuvants work. Today, in contrast, we are in a vastly different situation, and the science is advancing rapidly on a remarkable trajectory. Thus, I will speak over the next few minutes in the context of currently available scientific advances and technologies that have provided opportunities in the field of vaccinology that would have been almost unimaginable 10 years ago. The overall theme of my discussion will be the challenge of developing vaccines against microbes for which the natural immune response to infection itself is not completely adequate to control or eliminate the pathogen in question. So let me explain. For so many of the important pathogens for which effective vaccines have been developed, such as smallpox, measles, and polio, among others, despite varying degrees of morbidity and mortality associated with these pathogens, at the end of the day, the vast majority of individuals will clear the infection and will make an immune response that would then give them usually lifelong protection. So under these circumstances, our immune system has already provided us with a proof of concept that it is indeed capable of generating a protective response. And this has led to a fundamental tenet of vaccinology about which we're all familiar, namely, that the best way to develop an effective vaccine is to design your candidate to mimic natural infection. In other words, try to induce responses akin to natural immunity. This is usually accomplished by the use of inactivated or attenuated whole microbes. However, there are certain circumstances where natural immunity does not confer adequate protection against the pathogen. Some well-known examples are HIV, malaria, tuberculosis, and influenza. And so in these cases, we find ourselves faced with the challenge of designing a vaccine that will induce unnatural immunity. By this I mean an immune response that natural infection either does not elicit at all or does so only rarely, inconsistently, or weakly. In order to accomplish this challenging task, vaccinologists are employing, or should be employing, a variety of recently developed cutting-edge scientific tools. I will give examples of the utilization of some of these tools in the context of specific vaccines. So let's first start with HIV. This is a unique example of a virus to which, except for rare circumstances, the body does not make an adequate immune response. There are essentially no examples of a person with established HIV infection where the virus was completely cleared from the body by the immune system. Curiously, Neutralizing antibodies to HIV are not dominant among HIV-infected individuals in general, and when they occur, they do so later in the course of infection and are not effective against the currently circulating isolates in the body. The important neutralizing epitopes of the HIV envelope are often shielded by glycans that apparently render them inaccessible to immune recognition, despite the fact that preformed neutralizing antibodies can recognize the epitope. Thus, natural immunity does not seem to work with HIV infection. Early on, it was felt that the body was incapable of producing these antibodies, and only a few monoclonal antibodies that were broadly neutralizing were derived from the B cells of rare HIV-infected patients. However, with the utilization of extremely high-throughput screening of B-cell clones derived from HIV-infected individuals and rapid sequencing of their immunoglobulin genes, technologies that were not available years ago, it became clear that most patients can indeed make these broadly neutralizing antibodies. Unfortunately, they do so very weakly and too late in the course of infection. In this regard, by screening tens of millions of B-cell clones with precise HIV molecular probes, researchers have now identified human monoclonal antibodies that target the highly conserved, vulnerable structures on the HIV envelope and show exceptional breadth of neutralization, covering more than 90% of naturally circulating strains. In addition, investigators have used modern crystallographic techniques to determine the precise conformation of the epitopes to which neutralizing antibodies bind, and are using this knowledge to design much improved vaccines. The challenge now is to scaffold these epitopes to assume immunogenic forms that will induce the relevant antibodies. This is a classic example of structure-based vaccine design that hopefully will induce unnatural immunity to HIV. Another application of cutting-edge technologies that may allow us to develop candidates that improve the natural responses to infection is for influenza. Despite repeated exposures to both influenza infections and influenza vaccinations, most people are not protected against all emerging strains. One potential explanation for this is that in natural infection or vaccination, the virus does not readily expose those components of its structure that do not vary from strain to strain. However, some individuals are immune to subtypes of influenza viruses to which they have not previously been exposed through natural infection or immunization. This protection, which is termed heterosubtypic immunity, suggests that regions of the virus shared by different strains can indeed be recognized by the immune system, however, not very readily or consistently. It has been proposed that certain components of the virus, such as the hemagglutinin or HA, the nucleoprotein or the M2 protein, may be the targets for heterosubtypic immunity. So let us consider for now just the HA protein, the viral spike that mediates attachment of the virus to its target cell, which, as you know, is composed of two components, a head and a stem. The major immunodominant region of the virus resides in the head of the viral spike, which is multivalent and highly exposed to immune system recognition. The stem region of the spike is surrounded by adjacent HAs on the surface of the virus, which may actually shield it from recognition by the immune system and hence interfere with the generation of a natural immune response against it. Thus, natural infection and/or vaccination readily elicit specific neutralizing antibodies against the head of HA. However, anti-stem antibodies are not readily elicited. And so, by screening millions of antibodies from B cells, similar to what I just described for HIV, it has become clear that the immune system is in fact capable of making anti-stem antibodies, but only inefficiently. In order to overcome the shielding of the stem region from the immune system as occurs when the crowded HAs are presented on the surface of an intact virus, gene-based vaccine design was employed by Gary Nabel and his colleagues using a plasmid DNA expressing only the HA protein as a prime, followed by seasonal vaccine or HA adenoviral vector boost. This approach resulted in the production of HA stem antibodies in mice and ferrets with a vaccine from a 1999 strain that actually cross-neutralized not only itself, but also H1N1 virus strains from 1934 through 2007. It even generated neutralization of more distant H2N2 and H5N1. And so here again, with influenza, unnatural immunity can be successfully induced. In addition to influenza, gene-based and vector-based vaccine designs that present epitopes in their pure form, unencumbered by confounding molecules, have enormous potential in inducing immune response to components of other microbes besides influenza, where natural infection does not present the relevant epitopes in a favorable immunogenic form. Let's now very briefly consider the systems biology approach to modern-day vaccinology. Systems biology can simply be defined as an interdisciplinary approach that describes the complex interaction between all parts in a biological system with a view to elucidating new biological rules capable of predicting the behavior of the entire biological system. Since the response to vaccination in general depends on a wide array of interdigitating molecular, genetic, environmental, and other factors, the utilization of systems biology approach to the analysis of vaccines seems particularly appropriate. All of these data ultimately require complex analysis and computational models unavailable years ago that now would allow us to predict whether a candidate vaccine will lead to protective responses. And so it seems to me that if there's one disease where vaccine development is actually begging for a systems biology approach, it is tuberculosis. We know so little about the immune response to the microbe. We do not know why some people contain their latent TB infection, whereas others reactivate it. We have no idea why BCG induces the relatively restricted protection that it does. Clearly, a more comprehensive and integrated approach to the problem is needed, and a systems biology approach seems particularly suited to this task. In this regard, recent studies using microarray techniques to delineate gene expression patterns identify those cellular genes that were associated with active TB infection versus latency. A word about vaccine adjuvants. There has been some reluctance, particularly in the United States, to readily employ adjuvants in the development and deployment of vaccines. This seems curious, since microbes themselves express an array of adjuvant properties, particularly in their well-described interaction with toll-like receptors. In the spirit of mimicking natural infection in our vaccine strategies, it seems reasonable to fully explore the potential of adjuvants. Clearly, The use of adjuvants already has played an important role in the success of a number of important vaccines, including the RTSS malaria vaccine and various influenza vaccines, particularly those employed in Europe. Development of novel adjuvants, as well as the employment of existing adjuvants, should be aggressively pursued by vaccinologists. Finally, an important obstacle faced by vaccinologists is the long duration and expense of clinical trials. The typical approach to the development of a vaccine is the sequential approach to clinical trials. In this setting, a single hypothesis is tested, and if validated, proceeds to the next phase of testing. The problem is that if the trial is not successful, one usually returns to square one to test the next hypothesis. This approach is untenable for diseases such as HIV, tuberculosis, and malaria, where the incidence of disease makes it imperative that we develop a useful vaccine within a reasonable time frame. In this regard, I and my colleagues, including Larry Corey, Gary Nabel, and others, have recently addressed the issue of adaptive trial design in the context of HIV vaccine development. This concept is based on the principle of testing several hypotheses in parallel and allowing modification of the trial in response to data acquired during the course of the study. One or more decision points are built into the trial based on interim results. In essence, adaptive clinical trial designs can accelerate vaccine development by rapidly screening out poor vaccines while extending the evaluation of efficacious ones, improving the characterization of promising vaccine candidates, and identifying the correlates of immune protection. So in summary, I have briefly outlined for you selected aspects of the state of the science as they pertain to vaccinology. This is an extremely exciting time in the history of our discipline, since the scientific opportunities are almost limitless. The successes of vaccines have been breathtaking, and they stand alone in the enormity of impact that historically they have had on global health. However, important challenges remain, particularly with some of the most recalcitrant microbial killers. We now have at our disposal the scientific toolkit to meet these challenges and to elevate the field of vaccinology to unprecedented heights. From a scientific standpoint, it is up to us to thrust this field squarely into the 21st century. Thank you.